So here we are. Okay. So I'll offer that great architecture creates the spatial presence to inspire the soul. It provides shelter, comfort, safety, supports productivity. It can promote health, and it can supply more energy than it consumes. Great architecture can connect us with the outdoors while sheltering us from its extremes. It can be filled with the light of day or soak up the dark of night. And there are some places, when you're in them, you feel as if they were made just for you. Often in these spaces, you feel as much outside as you are inside. So today I'd like to posit a little bit about a topic that maybe you haven't heard too much about, the microbiome. And I think I have to talk a little bit about what is a microbiome and then try to convince you that you should care. And so I'll jump right to the punchline, which is daylight impacts the indoor microbiome, and the indoor microbiome impacts our human health. The exactitude of those mechanisms and their relationships have yet to be understood, and I think this is hopefully the beginning of a decade or more of research that may be required, as we've experienced in the last 2 decades on our IPRGCs, to begin to understand how they are impacting our health. I hope that this is the beginning of something along those lines. So I'll also say that as daylighting designers, we're all healthcare providers, or at least all part of a healthcare providing team. We've heard about that in various ways. Today, we know that daylight impacts visibility, visual comfort, glare, and that these things can be related to stress, provides connection to views and to the outdoor environment, and that this can in turn create micro-restorative experiences. It's our biggest sledgehammer, as we heard earlier, in terms of circadian exposure, which impacts mood alertness, melatonin suppression, sleep regulation, and it can also break down indoor chemicals through photocatalytic oxidation to modulate indoor air quality. And I'll show today how it also implicates the indoor microbiome. So we are all made up of trillions of microbes. Our guts, everything from our gut to the Mars rover is loaded with microbial communities. We can't sterilize this. NASA has given up. But what we can do is use it to our advantage and understand that the indoor microbiome, just like our gut, could implicate our health, our well-being, our mood. Did you know that about 5 kilograms of you humans, is not human? That by cell count, you are more non-human than you are human, and humans comprise about 70 trillion cells. We eat about 20 billion bacteria cells per day. We inhale about 3 billion, and we drink about 40 million. Everything from our skin to the Mars rover is loaded, and these indoor microbial landscapes might be in or out of sync with our personal microbial communities. And the slide, it would be a lot better if this animation were playing. If the provider in the other side of this room could click on the button to make this go, that would be delightful. Well, I will try it, and it's not going to work, which is really sad. I'll try one more time. If it's possible to click the button. Learning for next time to make sure this all works ahead of time. All right. I tell you what, if you're interested in that video, I will share it with you after on my laptop. It works brilliantly. All right. So what I'm going to show you next is a little bit of a deeper dive into that from a building at the University of Oregon. This is the Lillis Business Complex. And the first thing that we started to do at the Biology and Built Environment Center was try to understand how architectural factors like natural ventilation, night flush cooling implicate indoor air quality, because it seemed like the most direct measure. And so what you're gonna see here is that all of the offices in this space have a particular microbial signature. And if you were able to zoom in on that, you would see that offices are loaded with alpha proteobacteria, but are relatively, has a paucity of bacilli. Zooming in. So each of these spaces have a unique signature, and I'll just highlight a couple of okay, all of the animations are broken. So what you would see if I could highlight the classrooms for you is that they have also a very distinct microbial signature, and that bathrooms are distinct as well. So what we did next was look at the role of outdoor air. And while the animation doesn't work, at least this highlights, which is wonderful. The blue spectrum is what we did when manipulated the classrooms to be closed to outdoor ventilation. And so all they're getting is the resident microbial soup that we're all providing to the space as our contribution to science in this case. And that when we compare that to the rooms that are, that were ventilated with outdoor, you would see that it follows a similar line, but then as the outdoor ventilation takes hold over time, that the indoor community, the indoor bacterial community begins to look a lot like the outdoor bacterial community. And then it's reduced in abundance of human pathogenic related microbes. So this is not new in a way, but it's sort of new again. In the mid-1800s, we had culture-based studies that revealed bactericidal effects of light. And Downs and Blunt with their seminal study in 1877, and a series of studies following it all the way up until about 1950, which reinforced this idea using culture-based methods that daylight or any other sort of light action spectrum could impact indoor bacterial communities. So what we wanted to do at first was try to recreate the past. So we published a paper, sort of the first in about 70 years that we've found, that started to document this in more controlled situations. So we created these little microcosms. They're daylight models for the architects in the room who build daylight models, except they're hermetically sealed and they're also climatically controlled to be maintained at similar temperatures and relative humidities as what we would find in typical indoor environments. And then we, of course, measured ultraviolet light, invisible light. And we wanted to study, introduce this actor here. This is Pseudomonas aeruginosa. It's a gram-negative bacteria, highly antibiotic resistant, and known to be related to hospital-associated infection, in particular sepsis, and ventilator-associated pneumonia. So this is our actor, and we used controlled studies to plate Pseudomonas aeruginosa on these petri dishes. And then we exposed it over time to daylight dose in those microcosms. And you can see that the yellow line is the visible light, and it has an inversely proportional relationship to the survivability factor of Pseudomonas aeruginosa. And then on the purple line is ultraviolet light in those small microcosms, and again, inversely proportionate to the survivable factor of Pseudomonas. So the next piece, which is really bringing this 70 years fast forward, is that with modern DNA sequencing technologies, we can use things like metagenomics and metatranscriptomics and 16S RNA extractions to understand bacterial life in a way that we've never been able to before, because only 1% of bacterial life is culturable on average. And so with these new tools, we're able to have a much richer understanding of the microbial community. So in this study, we looked specifically at dust. And this just shows the spectra of the two different glass sources. And then we also did a dark covering. So we have a UVA, UVB, terrestrial UV enriched source, and we have a visible and IR enriched source, maybe more similar to what we might find in our indoor in buildings. And we use two techniques that, again, I'm an architect, so I have to lean on my friends in microbiology to understand these, but one is called Propydium monoazide, and it's a chemical, kind of a wet lab biological treatment of the dust, which will separate the total bacterial community from the viable, so it attacks the cell wall of bacterial community, bacteria that are not viable. And then the other thing that we use is something called qPCR, or quantitative polymers chain reaction, which allows us to count the total number of bacteria. So many studies today, even still with these advanced techniques, are just giving relative abundances. So with that technique, we can understand total absolute abundance. All right, to skip forward to a couple of the takeaways from this. So patterns in bacterial abundances were determined in part by the exposure to light, regardless of wavelength, in terms of the total bacterial community structure, and had a big impact. The largest difference in total bacterial community structure was between any of the bacteria in either of the light sources compared to the bacteria in the dark. This looks at the absolute abundances in a box plot. And the big takeaway here that I'll point out is that the two bars to the farthest left are the dark community, and all of the other bars are either of the light, the visible light being the yellow and the ultraviolet light being in the blue. And then the split in the graphs, or all the box plots to the right of one another, are the viable, and to the left is the total. And so that makes sense that the viable portion of each community is less than the total, right? But the big difference here, of course, is that there's much less viable bacteria in either of the communities exposed to daylight. This is another way to look at the data, in particular here, the community structure. And the more saturated colors are the total community, and the lighter colors are just looking at the viable parts of the community. In the total community, what you're going to see is a stark separation in bacterial structure from, again, what was in the dark to either of the light treatments. And then it also holds true in the viable portions of the community. But the types of bacteria that remained viable shifted, which is why you're seeing that offset. One more way to look at that is to do these violin plots. The more saturated version of each color is still the total, and the dark gray is still the dark community. And what we're seeing here is that dark total bacterial community looks a lot more like humans, in particular human skin, and that the light treatment communities look more like bacteria that survive outdoors, which is perhaps not surprising. But the kind of exciting piece, the part that might lead to health outcome kind of relationships, which we're not ready to describe just yet today, but you'll see that Saccharopolyspora, for example, is one taxa of bacteria that was of greatest abundance. It was the overwhelming bacteria in the dark communities. Again, these all started the same, right? So something is either blooming in the dark, which we're not completely sure of, or probably more likely being suppressed with the treatment of light. And so Saccharopolyspora has been associated with buildings in rural areas as well as environmental mediated respiratory disease. So that's the closest thing in this first study that we were able to come to that indicated perhaps a human health outcome. So I'll just wrap by saying I think this is the beginning of a new aspect, or a new again, an aspect that's new again about daylight. We've always known this bactericidal effect. We have new tools to study it. And I'd just like to thank the VELUX for inviting me, and of course the Alfred P. Sloan Foundation for funding the work.